Okay, welcome to the Bioinformatics seminar series. Today, I'd like all of you to help welcome Rachel Schwartz from the University of Rhode Island. She's going to be talking to us today about some of her work from her new lab, relatively new lab, looking at evolution from a computational perspective and kind of a methods development sort of perspective. So we'll be seeing kind of some of the new stuff that her own students have generated in the lab. So I'm looking forward to it. So welcome.
You can sort of take that into, oh, there you go. So this is work, some of it's what I did as a postdoc, but a lot of it's what my students have done over the last year and a half. So I'm super excited. We don't have a lot of definite results yet, but I'm kind of telling you where we're going. I'll preface this by saying I'm an evolutionary biologist coming into methods development rather than a computer scientist coming into genomics. So all of this is sort of from the lens of evolutionary biology on a whole variety of timescales. So this is the med school, so I figured maybe I better sort of point you in the direction of evolution and why it's so important. So this is a quote that probably most biologists put in their talk at some point. And the idea that nothing really makes sense except in the light of evolution. So all of the things that we think about, including medicine, you can look at with an evolutionary lens. And so you can think, well, we have a microbiome that we're interested in or interested in studying and interested in dealing with for our health. That comes from an evolutionary perspective, right? These microbes evolved to be symbiotic. You can think about your physiology as a product of our evolutionary history. And you can even think about our behavior this way, how we act as a product of our evolution. If you don't want to kind of get into thinking in these broad terms, this is a phylogeny. If you're not familiar with it, it's how we basically think about evolution, whether you're talking about these viruses here or you're talking about the whole tree of life. And from a sort of human health perspective, a lot of people do use phylogenies to understand host switching in viruses or parasites. So here in red here, this isn't my work, but it's a nice example. The MERS coronavirus, we might ask, where does that come from? And then we might extend that to say, well, if we know that the MERS coronavirus is closely related to viruses in camels or viruses in bats, do we expect to see similar host switches in viruses in the future? And I think that tends to be true, that bats tend to share their coronaviruses with us. So there's another one up here. This is the SARS coronavirus. And again, you can see it's closely related to the bat coronavirus. So there's a very direct application of phylogeny. I don't tend to think at that time scale. I tend to think at much larger time scales. So for example, how do we think about the evolution of butterflies? So there's butterflies up here. This is a monophyletic clade inside of all the moths and butterflies. So I think about this time scale. Or even at kind of this time scale. So this is all metazoans. And this is one of the questions that has emerged in the last 8-10 years is, what is the group that is sister to all other metazoans? So you have, is it the sponges? I don't know about you guys, but when I was in high school or so, they told us that sponges were sister to all other metazoans. And then another group came along, I think this was out of Casey Dunn's lab, and said, no, ctenophores are sister to all other metazoans. And then depending on how you do your analyses, you come up with sponges being paraphyletic and cnidarians and ctenophores in here. So we're still trying to understand how species are related, which it kind of boggles my mind sometimes because you'd think for a lot of these groups we'd have this all figured out by now, but we really don't. And so, like I said, I think about evolution at a whole variety of timescales. So we can think about sort of all of metazoans or even all of the tree of life, but we can also narrow this down. And I like to think about cancer as just very small scale rapid evolution of, I guess, populations really more than species. So here you'll see that this is somewhat similar to the phylogeny that I just showed you. So you have a bunch of individuals, and out of those individuals, you have one individual that goes somewhere else and evolves into a new population or new species, depending on sort of what time scale you're thinking about it. So cancer at the small scale, all the way up to evolution at the large scale, we need some way to understand how these cells or how these species are related. So I want to take you through a little bit about how we're thinking about getting the data to build phylogenies, or at least some of the challenges first, and then what are the methods. And I'll illustrate this with a great example in birds. People really like birds. They're pretty. They have all this phenotypic diversity, and so people get really excited about how did all this phenotypic diversity evolve. And so in 2008, this study came out, and this was really sort of the first big phylogenomic study. And they said, and you know, in 2008, this was genomic. We have 32,000 KB, 19 loci, and this was published in Science, and they said, we have figured out the relationships among groups of birds. End of story, we did it. There it is. You don't have to know the relationships among birds, but they do say, like, here are some of the historical relationships. That's what these colors are. Here's their relationships, and they're like, we're done. And they do have a really nice large data set. Say this. Unfortunately, if you look at different data sets, you still get different phylogenies. So there was your 19 loci. They've upgraded in 10 years, not even 10 years, 7 years. So now we have 12,000 loci. Pretty much most of the genome, the parts that they're able to align. They've only got 48 species, because if you've ever done whole genome assembly and alignment, that's a lot of work. So they're limited on the number of species. But that's a lot of data. And then they said, no, we have the relationship among birds. We're done. End of story. We have whole genomes. That's it. You can imagine that that's not really the end of the story. There's a third space there. And along came another group, and they said, well, you guys only had 48 species. We don't have as many loci, but we have a whole lot more species. And therefore, you know, you're not accounting for some of those really long branches. So we have the final answer for the bird relationships. So if you take a look at what these look like, and you can think about both of these are very large data sets, a lot of them are pretty similar, right? That's these horizontal lines, right? They're showing very similar relationships. And then you have things here. The flamingos and grebes here are sort of in this in-group. Versus here, they're closely related to pigeons and sandgrouse. So you see some pretty dramatic changes, thinking that they're related to the tropic birds and penguins and herons compared to pigeons and sandgrouse up there. So a lot of them are the same, but there's some pretty dramatic differences. And we don't really know what the right answer yet. I was trying to think of other cases where you have very, very large data sets and people get wildly different answers. We come up with a lot of data for that, but we're still arguing and we still don't really have a right answer. And the problem with this is also that getting a new answer, even if you're not sure that it's right, is very expensive. So if you've ever done whole genome assemblies and alignments, they're time consuming to do the sequencing and the assembly. They're expensive, right? This is a very long list of people that participated in this project, and they actually designed whole new methods to build phylogenies out of whole genome data. So it's a pretty big deal, and we still don't really know if we have the right answer. So this is kind of the answer that I like to think of. It's like work smarter, right? And this is what everybody wants to do. You want to spend less time on your project and less money and get the right answer. So this is the first thing is, for for me to understand getting the right answer in phylogenetics, we need to have the capacity to get large phylogenetic data sets more rapidly and with less money. So I'll sort of go through one of the processes that people have been doing to kind of shortcut this, and this is similar to what what the second targeted locus paper did, and then I'll explain how we're trying to shortcut this process. So if you have 5 genomes for which you have assembled the whole genome, or at least a reasonable draft genome, you take these genomes and you do an alignment. And we pull out the parts of the genome that are common. So this is the idea is you get orthologs this way. So these are the parts that tell you something about evolutionary history. And they have enough of a common sequence that you can align them, and then you can understand, what changes have occurred. So that's fine. It's not great for 5 genomes. You still have to assemble 5 genomes, but it's not terrible. It doesn't really scale up. So doing 48 genomes is obviously harder than doing 5 genomes. It's hard to account for rearrangements, for chromosomal fusions, for all the other things that are going on in the genome. But this is the approach that's taken by this group that works on ultraconserved elements to develop markers for phylogenetics. So they do pull out a lot of markers, but you still have to do the assembly and the alignment for those 5 genomes. So they shortcut it to some extent, which is how they get the target list. It's also the approach that's taken by what's called anchored phylogenomics. They sort of custom design markers for particular groups of organisms. The UCEs are trying to get markers that are more general, for example, for birds. I looked at the UCE website, and they say, these markers work really well. We've tested them. We don't really know what they are, but they work. So just don't worry about it, right? Which is kind of what phylogeneticists do a lot of times. Sorry, we tend to ignore the function of things. I'll get there in a minute. So that's fine. I know a lot of you work on humans, so you may have forgotten how hard it is to get a really good genome sequence for a non-model organism. This is the sort of the undergrad version of talking about genomes, right? I tell students it's, you have a genome, it's like a book, right? It's got chapters. Yeah, but when you try to sequence it, you're looking at taking all those books and throwing them in the paper shredder and then trying to figure out how you put it all back together. So this is the non-model organism. It takes a lot of time to develop a polished genome that you can really work with. The thing about this is that from my point of view, I don't really need the whole assembled genome, right? I want these core sequences. I want orthologs that tell me something about evolutionary history. So that's the idea, is how do we just get the core? How do we get those orthologs without going through the whole assembly and alignment process? So this is what I did as a postdoc, and we're still sort of developing the software as we add more pieces to it over time. And the basic idea here is that you have a whole bunch of reads. You might have tiny little short reads there, but you can figure they're usually 100 base pairs or 200 base pairs. And we take all of them, and we assemble them all together. And our assembly, we figure, looks something like this. Maybe you don't throw them all in. Maybe you just subsample them. The assembly might look like this. So there's your de Bruijn graph. If you're familiar with de Bruijn graph genome assemblies, you have parts of the genome like this that are have no variation. You have parts of the genome here that have a lot of variation, and you have a part over here that's maybe got a little bit of variation. So the parts that we're interested in from the point of view of phylogenetics are the parts that you could still align. Maybe they don't have this much variation, but you have some variation, and you're able to kind of trace that history back. So when your genome assembler spits out its results, it should spit out something like this. So We have a part of the genome where everything, all your species align to it pretty well. And that gives you something like this, where you get that locus for each species, but there's no variation. And then you have parts of the genome where they're so variable that they align, that they just assemble separately, right? Your assembler thinks this is just two entirely different pieces of the genome. They assemble separately, and then, of course, your individual species will align separately here and here. So you get both of the pieces back, but you don't really know that those are orthologous pieces. So they're not really very usable for phylogenetics. And then over here, you have that piece of the genome where it assembles together. Maybe it picks one of the alleles from the heterozygote, and you're able to align your data from both species back to that. And so it looks something like this from your two different species, and you get both sequences, the sequences from each species that you can then align together. So you know what the ortholog is. Doesn't really matter where it is in the genome. You know that that's an ortholog, and you know that it has variation, and you're able to see the alleles for each species separately. So it looks something like this. We take all of our sequencing, we throw it into assembler. You get kind of this composite genome. It's a hybrid. You take your reads from your individual species, align those back, get your genotypes, your alleles for each of your individual species, get rid of some of the extraneous stuff where you don't have enough data to do phylogenetics, and you end up with an alignment. So you have data for each species, for each locus that you're working with in order to build your phylogeny. So this is all one pipeline we've developed, and it was published in 2015. I call it SISRS. And so, yeah, so we have a piece of software. All you need to start with is your raw data that comes off the sequencer, or I guess your trimmed data, really. We provide some recommendations for trimming. But you take your raw data, you run it through the pipeline, and it produces a whole alignment, mostly just of the variable sites, because there's just so much data to work with. But you can pull out the loci as well. So there we go. So we have a pipeline. And as part of pipeline development, you have to show that it works. So it works. So here's a phylogeny of mammals. Tend to use mammals for a lot of things because there's a lot of data for mammals. So there we are. We have very high bootstrap support for these groups. Here you can see sort of one, or maybe the most accepted hypothesis for the sister clade to all other eutherian mammals. So there we go, right? I can show you it works for other species too. So this was a project I worked on using scissors to understand the evolution of Leishmania. So you can think about this as have a longer time scale applied to sort of understanding how the Leishmania parasite has evolved across different continents, and what time frame that happened. Did it jump continents, or are we looking at an older time scale for this evolution? So I made it sound all very nice. Scissors ends up being really fast, so we've shortcut this whole assembly and alignment process. What I haven't done with scissors is tell you that I have phylogenetic data that gives you the right answer. Like I said, we don't really know the right answer yet. We'll get there in a minute. So what this is another subset of the data supporting one of the other alternate hypotheses for mammals. So there's three major hypotheses about the sister clade to sort of the main group of eutherian mammals. In this case, the sister clade comes out to be just the Afrotheria, and then the sloth and the armadillo are sister to the remaining mammals here. So it's kind of good and it's kind of bad, because I can't tell you that we have the right hypothesis from scissors, or the right answer from scissors data yet, but I can tell you that scissors data outputs results that will give all of support for all of the hypotheses. So we know that the data that's in there, there's something about the data where different data sets are supporting the different hypotheses. So it's not that somebody's wrong. There's something going on with the data that we need to kind of work out. So this is what my lab is really interested in right now. I'd say this is sort of the meat and potatoes of the lab, and then we're starting to look at these loci more in depth. So where do you have signal in the loci for how species are related, and where do you have, and I shouldn't have said noise here, I need to fix this. It's really non-phylogenetic signal. It is signal not of how the species are related, but it's signal of how the genomes are evolving in some other way, whether it's convergence, whether it's incomplete lineage sorting, so sort of ancestral shared alleles. There are other reasons that we see this kind of non-phylogenetic signal, so it's not just noise. But we do want to sort it out if our focus is to understand species evolution. So this is where I get to talk about the stuff that my postdoc have been doing for the last year and a half or so. So we have a lot of data, and we have a really fast way of getting data for pretty much any group for which there's raw sequence data. So here's a phylogeny that we know pretty well. So now we can start to ask, out of all that SISRS data, which parts of the data tell us this answer, which we know to be pretty good, and which parts of the data tell us something that we know is not correct. So one thing that we've worked on a lot in phylogenetics, if you go back, say 20 years now, to the point where we were sequencing one gene or two genes and building phylogenies off of those, people have for a long time said, well, if you're looking at relationships kind of on this time scale within the last, couple million years, you would want to use a gene that has a fair amount of variation for that time scale. And that's going to be a fairly rapidly evolving gene, right? Because if you use a slowly evolving gene, it's not going to have any variation. On the other hand, if you're looking at this part of the phylogeny down here, or even older really, we want regions of the genome that are evolving relatively slowly, because if we pick regions of the genome that evolve fast, you're going to have multiple substitutions. And in the past, we've tried to model those and say, well, we can make sort of predictions about how many sites have multiple substitutions and where they are. You kind of want to avoid too many models and too many parameters. So in the past, people have said, well, we have a list of genes, and they actually give you a list. You go back in the books for genes evolving at the right rate to look at that part of the phylogeny. So we've known for a long time that parts of the data are useful and parts of the data are misleading. And now we've kind of said, well, we have the whole genome, so we're just going to forget all of that. We're just going to assume that the whole genome gives us the answer. But this is what we're starting to look at, is we take the whole genome, we take all our data for these known relationships, run it through SISRS, and so my postdoc's getting data that looks like this. So we have a lot of sites that map to our composite genome, so it's working pretty well. We have a lot of variation to work with, so that's a lot of data. And most of these sites are correct. And I'll show you what I mean by correct. So let me explain this figure a bit. So Bob's been working not only on primates, but on rodents and pecora, which are the ruminant mammals. And so he's able to find relationships, just like I showed you for primates, where he feels confident about those relationships and then look at which data supports which hypothesis. So what this is showing, if you just focus on these higher up blue parts of the violin plot, that's how much support there is in the SISRS data for relationships that we know to be correct. So each grouping of organisms is supported by a whole bunch of sites in our data. So that's good. So we feel like SISRS is producing data that allows us to estimate the phylogeny. There are some relationships, some splits between the data. So a single site will split your species into two groups. And so there's some relationships. You look over here. where there are sites supporting those relationships that we know are not correct. But it looks like most of the time those incorrect relationships are supported by a lot less data than a correct relationship. So we feel like SISRS is doing a good job providing sites that support the relationship that we expect in these groups. So now that we have a lot of data, and we feel like most of it's pretty good, but we're also supporting some of these relationships that, that aren't correct with some of our data, we can start to categorize the data. So because we're looking at primate data to start, we can annotate it. So you can start to say, well, we have a lot of sites. What are they? Are they CBS? Are they intergenic? Are they UTRs? Are they pseudogenes? Anything else? So we could put them in categories. He's just been working with these categories. We have a whole variety of other things that we're interested in. But we'll just start here. And this is, to go back to what I said before about certain regions of certain parts of the tree, we would think, well, we want faster sites, and certain parts of the tree we want slower sites. We sort of expect those faster and slower sites to line up with some of these categories. You expect coding sequence to be more slowly evolving because, for the most part, it's under a lot of evolutionary constraint. If you screw up the coding sequence, you don't get functional cells or functional people. On the other hand, pseudogenes, you would expect to see, it allows for a lot more mutations. Presumably introns or intergenic regions would allow for more mutations. So that's kind of what we're thinking, is that some of these categories are going to fall out in terms of more useful for either overall or in different parts of the tree. So it turns out this is partly true. So we definitely see different amounts of data supporting different parts of the tree. But it is surprisingly consistent across the tree which categories of data are being used for which parts of the tree. We're not quite sure if this is just we don't have a deep enough time scale. If you go back 100 million years, maybe we'll start to see different categories. So he's getting there, but we're not there yet. But this was really surprising to me because I figured that we'd have maybe more coding sequence back here. A coding sequence is this little sliver that you really can't see. There's not a lot of coding sequence in our data, which was really surprising. I thought that would be the stuff that assembled because it's evolutionarily much more conserved. Doesn't seem to assemble. And I think some of these are UTRs and some of them are intergenic regions. I forget what was what exactly. But yeah, this really surprised me that we didn't see these different categories sort of having different proportions in different places of the tree. So like I said, we have a lot of data. We're now having some results about we can get categories, we can start to get Go terms, we can start to think about how well we can model evolutionary change in each one of these loci. So I don't know about you, but if I have a known result and I have a lot of data that I can play with, I tend to think machine learning. Anybody else? Yeah. So I have a fantastic undergrad who is starting to do this kind of work, but she's an undergrad, so she has almost no time. So I don't have results from her yet, but she's starting to take the sort of the good sites and the bad sites and the sites at different time scales and say, can we make predictions about certain categories of data being more useful? So that's where we're going. Like I said, year 3 of me being at URI, so we're just getting going on this. But I'm super excited that she's starting to work on this project. All right, and so this is kind of where we're going, right? You want to pull the signal out of the noise, which is what machine learning is trying to do. Which categories of data are useful for phylogenetics and which are maybe providing more non-phylogenetic things. All right, so I'm going to switch gears a little bit. So I was talking a lot about just getting data in certain categories that is good or not as good for phylogenetics. I'm not, with that kind of project, I'm not worried about how those pieces of data are changing, how they're evolving, except for this one's evolving fast and this one's evolving slow, and maybe that makes it more useful. But one of the things that we're starting to work on in my lab is how are those genomes actually changing? And in particular, what kind of changes can occur in a genome and what kind of changes can't occur. So this figure is from my collaborator, Noah Daniels. We co-advised the student who's working on this project. I like to say he's sort of the flip side of what I do. I'm a biologist who tries to do a little computer science sometimes. He's a computer scientist who works in biology. This is not actually a biological data diagram, but it's where we'd like to go. So the idea here is this is a three-dimensional representation of multidimensional space, where if you think about evolution as, so in phylogenetics, we tend to think about tree space, all the possible trees. You can think about evolution as all the possible states in evolutionary space, so all the possible sort of ways that the genome could be, all the possible mutations, all the possible rearrangements. So you can think about all of that as like a giant landscape. What I'm interested in is how much of that landscape is occupied and how much can't really be occupied. So how much capacity is there for evolutionary change, and how much is evolution really constrained by selection? And when talking to my students, she's very interested in applying this to cancer, because if you think about a normal human cell, there's a lot of evolutionary constraints. That cell has a lot of functions. If you screw it up in a lot of ways, it won't work. Cancer genomes have all kinds of crazy things going on. They have rearrangements, and they have mutations, and they have deletions. So there's some variation, there's some mutations and structural variants in normal human cells, but there's way more in cancer. So we expect cancer genomes to occupy a lot more of that evolutionary space. On the other hand, they still have some constraints. So we're trying to see what sort of those underlying constraints are and how we understand them. So that's why the diagram that I showed you previously is that three-dimensional dimension reduction from this multi-dimensional space in an effort to visualize it. So my student, Yana, she's starting by looking at evolutionary space by using k-mer frequencies. So if you take your genome and you chop it up into little fragments and look at those patterns, all those little patterns are k-mers. And we know a lot about k-mers because they get used in metagenomics to distinguish species and communities in the microbiome, for example. So we know that k-mer presence and k-mer frequencies differ among species. That's been well established. You can see here that there's a lot of k-mers. This is for a human genome. There's a lot of k-mers that are very low frequency, and then there's a few over here that are high frequency. And so if we try and calculate what's changed between a sample of one set of k-mer frequencies and another set of k-mer frequencies, we can start to think about how genomes can change. And we're not quite there with cancer genomes, but we're trying to proof this with human genomes generally, because of course there's a lot of data for human genomes, and we know a lot about humans. So this is a population structure diagram for humans. And so you can see that there are populations that are quite cohesive. They don't show a lot of hybridization or mixing with other populations. And so if you think about these populations as sort of a way to ground truth, can you go back and say that these populations can be distinguished based on k-mer frequencies? So that's where we're going with our proof of concept. And the reason that we start to think that this might work is an undergrad in my lab for her summer project said, can we distinguish human populations simply based on k-mer, sort of the presence or absence of the most frequent k-mers. So the program MASH allowed her to just find the most frequent k-mers in all of her samples, and I sort of wasn't sure this was going to work, but I said, well, we need this comparison for what Jana's working on. And so she went through all these different k-mer values and said, can we find a k-mer value for which we can distinguish human population clusters? And it was kind of amazing, but she did. She found for a k value of 43, you get these nice dendrograms. So you look at the most frequent k-mers and create a distance matrix based on the distance between, based on presence absence of those k-mers. And from the distance matrix, you can create a dendrogram, and so we get population clustering, with the exception of this one lonely individual over here. But we'll just ignore that for now. So we have a sense that human populations do have frequent k-mers that allow us to distinguish them, which I thought was pretty cool. And it gives us kind of a baseline for saying that if the presence and absence of frequent k-mers can do this, we can probably do this with k-mer frequencies, sort of along our path to think about how genomes are changing. We want to be able to know that we can differentiate these groups. So Jana is using cosine distances to do these calculations. How do you calculate distances between a whole lot of k-mer frequencies? And one of the reasons she's doing this is that we're really hoping that we can, because of the way cosine distance is calculated, things that are very low frequency don't really matter in your distance calculation. And so we can get rid of a lot of the calculations that we would have to do to calculate the complete cosine distance. So if you think, you know, how many k-mers do you have when you have k equals even just 12? That's a lot of k-mers to calculate distance for. We'd like to get rid of some of them. Unfortunately, even getting rid of some of them, you still have a lot of calculations to do. So Jana was able to get this with k equals 12. We never really thought that k equals 12 was going to give us a great answer, and it doesn't. But it takes a lot of time if you're doing this serially. So because Jana's a computer science student, we said, go off and convert this to calculating distances on the GPUs. So that's what she's doing now, is developing methodology to do all of her distance calculations on GPUs so that we can scale up the k. So even with very large numbers of k-mers, she can start to do this. And of course, with human genomes, we have very large genomes, lots of k-mers. But she has an undergraduate who's working with her, who's trying to pull out, trying to use this approach on a smaller genome, namely for malaria, to look at fine-scale population structure, for example, in the Congo. So he's got a number of results like this. He's an undergrad, so the fact that he made this, I'm just happy with that. Yeah, because the little tiny lines and such. But there's sort of a group here, and then there's a group here, and there's a group here. So he's starting to use this approach, especially with the GPUs and the rapid calculations, to pull out the fine-scale population structure. And this is done, of course, straight from the raw Illumina data, right, or trimmed Illumina data, rather than trying to pull out SNPs or trying to look at microsatellites, so trying to process the data in some way. So we're trying to use this as a speed up. So I think that covers most of what I wanted to talk about. So I'll just summarize briefly. We have a method for getting a lot of data. We have the SISRS pipeline. It produces data. It doesn't necessarily, it's faster, but it doesn't necessarily give you the right answer. It gives you a lot of data to work with in order to understand what the right answer might be. So in order to parse out the data, to annotate it, to say, how do we determine categories of data, and then develop a methodology we can roll back into the SISRS software, where if somebody is putting raw data in and you have a sense of the time scale, or you have some kind of tree that you're starting to build, even if it's not right yet, that we could say, well, we're at this time scale, we're looking at these markers, we can categorize them and pull them out in a way that we can kind of filter this data to try and be more accurate about species relationships. And then we're trying to do this work on a more population scale to think about how do you use k-mers again, from raw data to be faster to think about populations, and ultimately to think about how genomes are changing and sort of how much evolutionary constraint we can see. Again, working with the raw data, trying to do this in a really efficient way that we can apply across a whole variety of data. So I'll conclude by mentioning all the fabulous people that I work with. I feel like I don't do any of the work anymore. My lab does all the work, and they're awesome. So Bob's been doing all the pulling out of data with SISRS and annotating it, and working with Caitlin to start thinking about machine learning. Yana is doing the population structure work with k-mers and all the GPU stuff and all the computer sciencey programming things that I really don't understand. And she's been working with Bayzat to produce the results from MASH based on presence and absence of k-mers, and then with Devin to think about population structure of malaria and applying her method to other cases. And of course, there's a lot of funding that goes into this. NSF funded our original SISRS work, and also funded Bob. He's on an NSF fellowship for the next couple of years, so we're excited about that. URI provided startup, and they provided funding for Yana for a summer, and they provided funding for the undergrads who've been in the lab. So with that, I will take any questions.
Any questions?
So different people makes different trees for the same data. That's a common problem. So is there a way to quantify the difference between different trees for the same alignment?
Yeah, so there's a number of methods to look at how different trees are. So there's distance metrics between trees. Robinson fold distance is kind of the standard method to look at how different they are. So that's sort of a distance metric, but then also the SISRS data for mammals, for example, supports the 3 hypotheses that we know of that are sort of well established for mammals. So it's not necessarily a distance thing, but the interest is in which is sort of the first thing to branch off, and which and what that group consists of. So sometimes it's distance between trees, and sometimes it's which hypothesis you're supporting.
So looking at like the different types of sequence and which ones are more or less helpful for getting accurate results, isn't it actually kind of not surprising that the coding sequence is just that small sliver, just because it's such a small fraction of the genome? Like, did you expect that, or you know?
So we were thinking that most of what would assemble is being conserved. So even though it's a fraction of the genome, that you wouldn't assemble a lot of the introns across this time scale. So you just would miss a lot of that data, and that a lot of what would be assembled would be the highly conserved data, which would be coding sequence. So that's why I was thinking it would be more of the genome. It may be at different time scales that we'll see that pattern, because these are primates, and if you go back 65 or 100 million years, we may start to only assemble coding sequence, but we haven't done that.
I guess along that same question, if you were to look at other species, do the markers you find also tend to match the distribution of where you would expect to find features in the genome?
So if you add, you mean just if you add more species?
Or if you look at like not just primates, what you would tend to expect to have like?
Oh yeah, so that's sort of another problem, and this is why I don't think we'll come up with just categories that are good and categories that are bad. I think we'll come up with categories that are good for certain timescales and categories that are good for certain clades, and those will differ. And so the sort of very simple thing is we'll do this for 1 or 2 clades, and we'll be able to report that for this clade or that clade, you use these markers. What we really are hoping to do is come up with a way of generalizing that. So if you get all your SISRS data and you can build kind of a preliminary tree, so you have kind of some sense of maybe some of the best supported relationships, and you can feed that back into a module in SISRS to say, okay, we know something about sort of our preliminary data. How do we then filter based on particularly the nodes that are well supported? So what categories at what time scales are supporting, I would call them easy nodes, nodes where there's a lot of distance between them and other nodes. And if you can understand the data that are supporting those nodes, maybe you have a chance of supporting the more complicated nodes, things that have short internode distances. So I don't think it'll be the same for every group, and what we really want is a way of having SISRS be able to filter for that particular clade or that particular time scale. And this may even be building different parts of the tree with different data, which I think is important, because if you have a large time scale and you can't really expect to build the shallow relationships and the deep relationships with the same data set, but that's what we tend to do. So how do you parse those things out?
So I have a couple questions too. So I think that this is a really great talk, and SISRS is really innovative in the way it works. I really like this idea that you just kind of brought up about, and I have a little question about that. So can you use maybe the number of species involved in the loops in the De Bruijn graph as kind of a prior for the tree calculation itself, and maybe in some sort of iterative process as you refine the branches of the tree, you're only looking at fewer species aligning to the De Bruijn graph in that location versus only focusing on regions where lots of species are actually aligning.
Yeah, so you would have parts of your graph that are 4 species, and you could say, so we kind of talked about this. We talked about having an assembler that would work well for SISRS, and we talked about having a colored De Bruijn graph where you're labeling the reads such that you can parse out, sort of things that go with particular species. So we've talked about a couple ways of approaching it, but we haven't really tackled that problem. I think it makes a lot of sense to look at the De Bruijn graph, but yeah, we haven't tackled that problem.
And then another kind of related question, I guess, is that it seems that in both the De Bruijn graph creation and in the k-mer calculations, repetitive elements are going to be very highly represented, because even within a single species, if you took the human genome, the parts of the De Bruijn graph that are more complex but still mappable are more than likely just from like line elements, for example. So you have lots of variation, lots of copies of them. And then the same thing with the k-mers, the low complexity repeats in the human genome are going to easily be the most common 12 k-mers that you're looking at.
That's a good question. I haven't looked at whether those, and maybe it'll come out when it's just something to point Bob at for another category, to say, are we assembling a lot of these repetitive elements, and do we need to filter them out, or do we need to handle them in some way? That's a good question. I haven't thought about those yet. As far as k-mer frequencies, the distances, so they would be the most frequent k-mers. I'm trying to think whether that's going to affect the distances between the species. So I sort of feel like that would be helpful, actually, because you're looking at the frequency of those k-mers. So that is what's changing, which is what we're trying to calculate the distance for.
Maybe more in the human population comparisons, though. So if you're looking at the differences between human populations, maybe some of these have been vastly expanded differently in different populations. But for the most part, it's going to be centromeric repeats, not the satellites that are the same generally.
Right. So you get, so there would just be very limited distances, I guess, in that case, because if you had 2 samples with k-mers from those repetitive regions, then the frequencies will be pretty consistent. So actually, one of the things that Yana and I sat down and talked last week before Thanksgiving, and I was like, maybe we should just look at what's changing and what isn't. What are the things that are changing dramatically between samples, and what are the k-mers that are common? So we haven't done that yet, but it's something I've thought about, is actually look at what those k-mers are.
Hi, really nice talk. So my question is along the same lines. So I'm guessing your answer is probably that you haven't looked yet. But in terms of the k-mers, I was wondering if you've looked at, well, first, your data was all genomic data, not ESTs. Is that correct?
Yep.
So
they're all
have you
whole
looked
genome.
at how many of them coming from coding regions or just expressed RNA regions versus intergenic regions? Because I would guess you might get fairly different answers depending on what you,
Yeah,
which
that's,
type of k-mers you use.
yeah. We have it. We've just been doing very broad brush genomics. But yeah, it'd be interesting to look at sort of different subsets of the data in that way and say what patterns do we see based on the subsets of the data that we're looking at. That's a really good point.
Yeah, and then one more question, if I can. One more. This is a much broader question, but I was thinking that oftentimes if you get an incorrect answer, I guess, evolutionarily, there still is probably some truth in it. You know, like some convergent evolutionary pressure, you know, on this. Have you looked into any of your kind of false positives to see what you might be able to learn from
yeah, so there's
species?
There's a lot of reasons, and that's why I said I have to correct myself and refer to it as non-phylogenetic signal, because it's not the relationships among the species, but it can be the relationships among the genes. It can be the relationships, sort of the historical allele incomplete lineage sorting. It could be convergence. So there's a number of reasons that we might have that non-phylogenetic signal. We haven't looked at whether that falls out into particular categories. Yeah, that's a good thing to kind of parse out in the end. Thanks.
Great. Let's thank Rachel again. Great talk.